Hello again, and welcome to our webinar. Good morning, good afternoon, and good evening to all of those joining us from all around the world. My name is Dusty Gallagher. I'm the Executive Director of the Phytobiomes Alliance, and we're really glad that you've joined us today for this great webinar that we're going to have, and we hope that you will make it interactive as we will have time for a Q&A after our featured presentation today. So just to begin, I would like to just give you a brief overview of the Phytobiomes Alliance. We are a nonprofit consortium of industry, academia, and governmental scientists, and our vision is that by 2050, that all the farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for determining the best combination of plants, management practices, and inputs for a specific field in a given year. We would like to take a moment to thank and provide a special thank you to all of those who sponsor the Alliance. Without that sponsorship, we aren't able to do the good work, and we wouldn't be having this webinar today. So thank you so much for your sponsorship and your support. So just a reminder, when we talk about the phytobiomes, we're talking about the full complex system of plant-based agriculture. So we have plants growing in a particular substrate. For the purpose of this graphic, it's soil. And then we know that there are a whole host of interactions that occur within the system. We have microbiomes, microbial activity in the soil. We have other animals and plants and insects that also provide some interaction. We have the influence of climate and weather. And then we also have all of this is influenced by the management practices that are chosen by the farmer. So it's this full complex system that we're talking about in the interactions among all of the components, between and among all of the components that we're talking about when we say the phytobiomes. And I would like to just introduce a couple of upcoming webinars that we're having. We already have 2 more scheduled this fall. On October 8th, we have a webinar organized by our microbiomes working group, and that registration is available in live. And then we have one on a month later on November 12th. And this one is organized by our animal microbiomes working group and is also available for registration. So please go ahead and register for these webinars. You will not be disappointed because they're going to provide another good hour of content. And then the Phytobiomes Alliance is also working to organize some workshops at the Plant and Animal Genome Conference in January of 2026 in San Diego. We will have the Exploring Phytobiomes workshop, as well as the uniqueness and commonalities between plant, animal, and soil microbiomes. And we are accepting abstracts for both of these workshops. If you're interested in showcasing your research work within the phytobiomes, please submit an abstract. That call is open, and you can see and submit your abstract with that link available there. So just a few housekeeping items today. This webinar is being recorded and will be available in about 24 hours post-webinar on our Alliance YouTube channel. And following our great presentation today, we will have about 10 minutes or so of Q&A. And so I would just like to remind you that we do have several features within the Zoom platform, and the Q&A panel is where we would like for you to submit those questions. The chat feature is available for you to see messages from the organizers. So we will be posting links, we will be posting information that is provided in the presentation in that chat panel, but it's only a one-way communication. It's not interactive. So please use the Q&A panel for your questions. And we will also be providing the PowerPoint slides, PDFs in the chat panel. And if you are not already on our mailing list, that is the best way to receive information about the Alliance, webinars, workshops, and information. So please go ahead and sign up to receive that news. So without further ado, I would like to introduce our speaker for today. Ilenne Del Valle is a research staff scientist with Oak Ridge National Laboratory here in the US, and she will be speaking today on designing biosensors to inform ecosystem engineering. So I would like to invite Ilenne to the screen. You will be able to share your presentation, and welcome.
Thank you, Dusty, for that wonderful introduction, and to all the organizers as well for the invitation to present today. I'm really excited to be here giving this webinar for the Phytobiomes Alliances. So today I'm going to be discussing some of the efforts that we have done trying to develop biosensors to inform ecosystem engineering. Before I jump into the presentation, though, I would like to talk a little bit about my workplace in Oak Ridge National Laboratory. So Oak Ridge is part of a network of 17 national laboratories here. You can see a picture of how our campus looks like in this slide, that are part of the US Department of Energy with a mission to advance research that addresses national priorities such as national security, climate, and energy. So as part of Oak Ridge National Laboratory, and more specifically as part of the biosciences division here, we conduct research to develop a circular bioeconomy. And a circular bioeconomy depends on reliable biomass production. So one of the big questions that we have here is how can we improve biofeedstock productivity? There are multiple ways to do this, but one attractive approach to do so is through microbiome engineering. So I'm sure this community is aware of this, but plants provide a variety of different health benefits to plants. For example, they can help them with acquiring nutrients, defend them against different pathogens, enhance stress tolerance, and overall improve soil health. So one alternative to do this is to use these so-called soil probiotics or microbial inoculants that can contain bacteria to augment these different properties, or even though they can contain engineered microbes that potentially can have functions that can help the plants grow better. Now, one of the challenges that we currently face with these type of technologies is that introduced microbes often fail to thrive and persist under these different environmental conditions. And there are a lot of different studies that have shown that when you are introducing these microbes that are not adapted to live in these different conditions, they quickly die off and they cannot provide the functional impact to the plant. And this is because the microbial establishment in the rhizosphere is a very complex process. So in this figure that I'm going to show here, we have divided them in different stages. So first of all, the microbe has to be able to establish. They have to be able to grow under these different soil conditions. Once they have grown and sustained their growth under these environmental parameters, they have to be able to spread and persist. And in order to do this, they have to coordinate their different behaviors as a community. Once they have done that, they can start interacting with the plants through plant-microbe interactions and communications in order to perform the functional impact. So one of the things that we want to do is that we want to discover key traits that impact this ecological process in the rhizosphere. So the overall objective of our research is to develop tools to understand control, but also monitor the establishment, spread, and functional impact of these introduced microbes. The work that I'm presenting here today and that I'm going to be describing primarily focuses on the tools that we are developing for both understanding and monitoring, and it's part of an effort of a science-focused area in the Biological Energy Research Department within the Department of Energy, and specifically within a science-focused area called Secure Ecosystem Engineering and Design, or CDSFA, which I'm gonna describe a little bit at the end of this presentation as well, in case you guys are interested in knowing more about these efforts. So in order to create these different tools, we need to design a pipeline for understanding engineered microbes and their behaviors in the environment. This pipeline overall looks something like what I'm describing in this figure, where we first need to isolate different microbes that either exist within the microbiome of our plant of interest, or alternatively, we also use microbes that are present in already commercially available products that are used as, for example, biopesticides or biofertilizers. Once we have these microbes, we have a pipeline in order to domesticate them, which includes different methods for introducing DNA into them, as well as creating different genetic circuits to make them function in different ways. And we do this through different design, build, test, and learn cycles in order to learn how they can maintain and improve upon those functions. After we have done that, we can use these genetic circuits in order to create genome-wide libraries in order to do something that we call genotype diversification. So we create different variants with different traits that we can later on test in the environment to see what the impact of these changing these genetic traits have on the overall establishment, persistence, and metabolic activity of these microbes. And then once we have these microbes, as I mentioned before, we can do these functional genomics studies where we see what are these plant-microbe interactions. So first, I'm going to briefly describe some of the efforts that we have done in order to domesticate these microbes of interest. And because we work at the National Lab, which is a very interdisciplinary group, one of the things that have really helped accelerate these advances and discoveries is actually using physical automation to streamline some of the workflows that are required for this microbial domestication and that they can be quite laborious and tedious. So I'm showing here how the labs actually look like. So we have, for example, this automation lab that has pipelines to increase the throughput of introducing genetic material into these bacterial probiotics. So we have nicknamed them actually Kirk and Spock because we are Trekkie fans, but basically one of these automation systems, which contains liquid handlers, electroporators, and other equipment that we require in order to engineer these microbes. One of them is a complete aerobe system, but then we have an equivalent one that is enclosed within an anaerobic chamber. So we use it to study anaerobe microbes as well. So using this platform, we can go from basically screening some of these genetic modifications that we are doing in genetic circuit design very rapidly. So if you try to do this by hand, maybe you can do maybe 10, 20 96 well plates of samples. So we are talking about, I don't know, 300 samples per day. These robots here allow us to basically screen more than 50,000 different samples per day. So it really accelerates the way that we are designing these different microbes. So I'm going to give an example of one of the workflows that we have automated using these different systems. So one of the challenges when you are trying to introduce genetic material in these different microbes is that they have defense mechanisms in order to evade the recombinant DNA from entering these cells. So when you have a synthetic genetic circuit and you want to engineer this bacteria, they have these systems called methyltransferase systems that contain restriction enzymes that cut this DNA. So one of the things that you can do is that you can automate a pipeline in order to sequence the genome, but also analyze the methylome of these bacteria, which contains different motifs that these restriction enzymes recognize and then they cut. So when you have information about the different sequences that are getting methylated and cut, you can basically create different mimics in E. coli that expresses these methylases and then produce the methylation pattern of your bacteria of interest. So when you isolate DNA from this bacteria, then it gets recognized by the host, and then they are kept and maintained there. So as an example, I'm showing here some data of one of the plant growth promoting bacteria that we are using. It's a type of Bacillus, not Bacillus subtilis, but a cousin of it called Bacillus velezensis that has been shown to have a lot of advantages in terms of producing different phytohormones and other secondary metabolites that help plant growth. So we have two different strains of these Bacillus velezensis, and when we have applied or automated pipeline for methylome analysis, we can see that the methylation motifs that they prefer, the ones that I'm showing right here, are very different, even though these two strains are they're very similar. And when you are trying to just introduce DNA from a E. coli cloning strain that contains the circuit of interest, you can see that if you are just isolating the DNA from the regular E. coli cloning strain, you get almost no transformation efficiency. Even if you use an unmethylated strain, so one that doesn't have any methylation at all in the DNA, you get poor transformation efficiency. However, when you are using the mimic E. coli or DNA that you have isolated from the bacteria itself, you rapidly increase the transformation efficiency of these microbes by several orders of magnitude. So in this case, up to 6 orders of magnitude. So our ability to engineer them rapidly increases, and we can screen multiple bacteria at the same time with multiple conditions using our automated platform. So once we have been able to introduce DNA, we can start testing different genetic parts to control, for example, transcription and translation of these different systems. Once we have done all that discovery cycles for understanding how we can express our genetic circuit better, we can apply them to do these genome-wide studies. So in order to do this, we require these new technologies in order to understand genotype to phenotype discoveries. So here I'm going to show some of the tools that some of the other groups that work within the SID SFA are also developing. So here, for example, if I have people in the audience that are plant biologists, they might be familiar with something called genome-wide association studies, or GWAS. These are highly effective techniques that are used in eukaryotic genetics that link genetic variation to phenotypic traits across natural and experimental segregating populations. However, they are mostly applied to macroscopic sexually recombining eukaryotes, such as humans and plants. And unfortunately, the use of this technique and also quantitative genetic mapping in bacteria has been limited by the lack of native systems for sexual recombination and generally low recombination rates within bacterial populations. So again, while these GWAS approaches have been successful in plants to understand how genetic variability can impact different traits, this is not so easy to perform in bacteria. However, Josh and his team, they developed a technique called genome shuffling, in which you can protoplast gram-positive bacteria and then make them fuse together so they can recombine and divide with a mix of the two genomes of the parental strains. So here I'm going to show some of the data and how it looks like for these genetic variants. So basically here we are showing all the genetic variants and recombinant progeny and how they have either black or blue depending on what part of the genome it's from one parent or the other. And what we can see is that these recombined regions were broadly distributed and included short inserts, so less than 100 base pairs, large multi-gene fragments of more than 60 kb, but they can also even include different single nucleotide polymorphisms, or also called SNPs, which vary averaging between 1 every 50 base pairs. So it gave us a wide distribution of different genetic modifications and within these recombinant progeny. So what you can do with this different panel is that you can start thinking about different traits that are important for establishment and persistence in the rhizosphere. So let's take, for example, these bacteria and how they need to survive the conditions of starvation, right? If it is a gram-positive bacteria, they can sporulate. And then once they have the nutrients back, they need to germinate back up. So you can take, for example, two parents that have different germination rates. Here showing once again in black and in blue. We have these two different species of Bacillus subtilis, RONN1 and the DK1042. And they germinate with different stimulants and at different rates. If we screen the progeny from this QTL mapping that I showed you in the previous slide, you can see that you get a mix and match of intermediate phenotypes of these bacteria. So then we can start mapping out what are the genetic modifications that cause this using the genetic information that we have from those mappings. So this is I'm sure some of you may be familiar with this, but this is called a Manhattan plot and basically summarize the statistical significance of the associations between each genetic variant along the genome and the spore germination across the QTL mapping population. These graphs tends to look a little bit more messy. So this one doesn't look quite as much as a Manhattan plot, but rather like a I don't know, something with a really large tower in there. But if you zoom in into this region over here, you can see that there is quite an association with one receptor here that seems to play a role in how these different bacteria germinate. So this is potentially a trait that we have characterized that we can potentially later on engineer into these plant inoculants if that is what we wanted. So if you guys have more, want to know more about this technique, it was recently published into bioRxiv. Another complementary strategy to create genetically diverse bacterial population is something that Carrie Eckert and her team works on, which is to develop genome-wide CRISPR-I or CRISPR interference libraries to create controllable knockdown levels. So just with our RNT-seq and other knockout techniques, you get a yes or no answer. However, there are a lot of different phenotypes in which you have variable levels of response. So let's take, for example, biofilm. If you produce too little biofilm, maybe the bacteria are not going to be able to attach to the roots. However, if you produce too much biofilm, you might trigger plant defenses. So you want something in the middle. So using these CRISPR knockdown libraries, we can start addressing some of those questions. The way that this works is that you basically design 10 guide RNAs per each gene in the genome of the bacteria. So in the case of some of these plant growth promoting bacteria that we have been working with, we are talking about designing around 40,000 different constructs that are going to produce these knockdown levels. Once we have them, we can grow them under the different selective conditions and create a fitness map that is going to tell us which genes are enriched and which genes are depleted. The genes that are depleted, it means that having them knocked down, it was a bad thing, so it means that they were essential. If they are enriched, it means that actually knocking them out was something beneficial. So you end up with data that looks like this. So here I'm showing, for example, two bacteria that were grown under low and high stress. And some of these genes and their patterns look different. So we can delve a little bit more into what these genes do and then how we can engineer them in order to, for example, make them adapt to stress more quickly or be more resilient. All right, so now that I have talked a little bit about how we are doing this genotype diversification in the bacteria, so then the next step is to basically validate these candidate traits. Do they work? Do they not? We have engineered them. There are some of them that they seem to be working well in the lab. Do they actually work well when we have the plant microbe and the complex environment in there? So I'm going to take a little bit of a step back and show some of the things that we have learned along the way when we have tried to validate some of these candidate traits. So for example, I'm going to show here something that we did trying to understand the role of biological communication and the outcome that it can have when we are trying to engineer these plant microbe interactions. So as you guys know, biological communication involves the production, release, and detection of small diffusible signals that trigger behavioral responses within or between organisms. This can be things like, for example, nodulation, or it can be something like producing antimicrobials. So they play a key role in modulating a lot of these plant microbe interactions. Now, one of the things that we have to take into consideration is that it's not so simple as to just produce more of these signals that are used for communication, or maybe produce less or produce molecules to disrupt them. Because the bioavailability of these signals in the environment can vary quite a lot. It depends on a lot of different things. For example, these signals can get consumed, they can get converted into other chemical forms, either by biotic or abiotic factors. They can get sorbed, they can change and maybe become not spatially available if you have a drought and there is less interconnectivity in the environment. So there is quite a little bit of complex interplay between soil properties, the signal that you are trying to study. So having said that, one of the ways that we think that is a very powerful tool to understand these signals in the environments is basically asking the microbes themselves to see what they are perceiving. And one way to do this is through the design of different biosensors. I'm sure, again, most of you have heard what biosensors are, but these are genetically engineered bacteria that sense an environmental input and then can translate that into a detectable signal that we can then later on measure. So there are two different types of biosensors that you can engineer. One of them is a constitutive biosensor that you can just use them in order to see how the metabolically active the cells are. This is a powerful tool, for example, in the case of some of these Bacillus strains that I was referring previously in my presentation, because if you just use metagenomic analysis, sometimes it's a little difficult to parse out whether if these signals came from live cells, dead cells, sporulated cells, and in reality, what we care about is basically whether if this fraction of cells were metabolically active. The other thing is that biosensors allow you to report in real time and constantly, rather than giving you snapshots that are disrupted. The second thing that you can do is that you can design an inducible biosensor for measuring the bioavailable fraction, of signals instead of the total chemical content. So for example, if you wanted to validate traits regarding biological communication, you could potentially use an inducible biosensor to see how the dynamics of communication are happening in the rhizosphere. So it sounds really nice when we presented it like this. It has a lot of advantages. It's a cheap tool, easy to engineer with these new advances. However, we have learned that it's not quite as straightforward to do. So I'm going to start with one example where we very naively tried to use biosensors in order to understand what was happening between plant microbe communication that was mediated from a family of compounds called flavonoids. So this is a family of low mass C6, C3, C6 plant secondary metabolites, and they play a really key role modulating a variety of different processes that are important in plant microbe interactions, like for example, chemotaxis, defense against pathogens, allelopathy, nodulation, etc. So one of the questions that we wanted to address is that, okay, if we have, for example, bacteria that have different ways of communicating, how do the soils affect this plant microbe communication that is mediated by flavonoids? So we made a very simple biosensor. It responded to one of these flavonoids called naringenin, and then it produced a fluorescent reporter that is really well-known and studied way of using these biosensors, using like a visual reporter. And we did some experiments in the lab with this. We will mix the flavonoids with the soils, and we use soils from 3 different sites and land usages. These included agricultural, meadow, and forest sites. And then we incubated them together, we discarded the soil, and then we added the biosensor, and we measure how much of the leftover naringenin that was there were the microbes actually sensing, and then correlated that bioavailable naringenin to the different soil properties. And what we found was something really interesting, and it was that as you increase the content of organic carbon that is present in your soil, we saw a decrease in the biosensor ability to sense this naringenin. So this suggested that somehow soil organic carbon was actually decreasing the flavonoid bioavailability. And we were a little surprised about these results because usually soil organic carbon is a good thing, right? And then the more, in this case, for example, this flavonoid plays a role in nodulation. So we expected that more nodulation, better. So it was a little bit confusing to us. So in order to determine what exactly was happening, we had to do a lot of different experiments. We did experiments with soil, with organic rich soil amendments, with biosensors. We did a lot of analytical chemistry like LCMS and MR to try to get at the mechanism of what was happening. Once we determined the mechanism by which the soil organic carbon was decreasing the bioavailability of this biological communication molecule, we did some plant microbe studies to try to see whether if the decrease was actually relevant or if it was just something that it happened and the threshold did not affect the actual outcome of the ecological interaction. Ultimately, after doing all those different experiments, we were able to determine that the polyphenols that are dissolving the organic carbon binds to the flavonoids, and they do reduce the signal transmission at an ecological scale, and that this had implication for different types of feedback that the environment was giving the plant and the microbes to modulate these plant microbe interactions. If you guys are more interested to know a little bit about the results from this study, you can look at this science paper that was published a couple of years ago. But again, we spend a lot of time trying to track down this one single mechanism. So we learned some things along the way, right? One of the things that we learned is that you cannot do these biosensor studies in situ directly because a lot of these soils that we were using were rich in carbon. They were very dense, opaque materials, and it was difficult to see these visual reporters from the microbes. Another thing that we learned was that it was really time consuming to characterize the quenching mechanism. So we needed to find ways in order to get at this mechanistic information so we could increase the throughput of these experiments. So how can we improve biosensors to study microbes within soils? That was the next question that we had. So again, I mentioned these biosensors translate the environmental signal into a visual output, a fluorescent reporter, a pigment. So we decided that instead of doing that, what we could do to improve this system was to use a biosensor that as an output, it produces an indicator gas. So this indicator gas can diffuse out of the soil, and then you can measure it in the headspace of samples without having to disrupt them, and then using analytical tools like, for example, gas chromatography, mass spectrometry. One of the advantages that this sensor had, besides being able to function within the soils, right, without having to disrupt the samples, is that you can use them at different scales as well. So environment ecologists in general, they have been measuring gases in the greenhouse and in the field for a really long time. So there is a lot of different analytical tools that you can use in order to measure these gases at different scales. So currently, one of the things that we are working on is actually translating some of the studies that we have done with these gas biosensors in small mesocosms in the lab to systems that are larger, like big soil columns using thermal desorption, or analysis in the greenhouse using things like PTRMS. So one of the advantages of these new type of biosensors, gas biosensors, is that allows for below ground microbial monitoring. So that's how we kind of like set up to fix one of the challenges that we had in the previous study. The second thing is that we needed technologies that we could develop to simplify the study of microbial processes in the soil. So again, we are not the only ones that have thought about this, and a lot of different researchers have developed different types of intermediate systems that you can use in order to get at these mechanisms that are a little bit more complex than just your liquid culture in the lab, but less complex than what you will find in the field or even in the greenhouse. So we use a variety of them. I, in my research group, work with several of them, including soil slurries, microfluidic devices. But today I'm going to talk about one of them, which is basically these silica-based synthetic soils that we can basically make and that can help us understand different individual drivers that are altering these microbial behaviors. So they kind of look like this. You can see them as they become more similar to the natural soil in these different vials, but we basically make them like a cooking recipe. You start with a mix of silica particles that have different particle size distribution to mimic different texture in the soil. You can add different minerals, you can change the pH, you can add different sources of organic matter, and then we have a method in order to aggregate them, so to make this microaggregate structure that is very important in soils using physical methods of wet drying cycles, and then we can load them with whatever nutrients we want. So one of the advantages of using some of these systems is that we can control each one of these parameters and then correlate some of these soil properties and how they're impacting the microbes. So, and as an example that I'm showing here with one of these first variables, if we are changing particle size distribution, we are of course changing gas and water movement through the soil column. And we can characterize that property using water retention curves in these artificial soils as well. So something that it's done in the field, it can be done in the lab with natural soils. We can also apply the same tools to characterize our synthetic soils. So one of the things that we did in order to test if the gas biosensors plus the artificial soil could make our life easier to understand this one trait that we wanted to validate was to apply it to understand how soils affect microbe-microbe communication. So microbial communication in the soil is also very important. They use it. Here I'm showing one example of this microbial communication that it's mediated by a signal that is called asyn-homosenine lactones, or AHLs for short. And they modulate a variety of different processes. For example, the transfer of genetic material, the production of greenhouse gases, the production of hydrolytic enzymes as well. So similar to before, we created a biosensor that, that is capable of sensing one of these signal molecules, a long chain AHL called 3-oxo-C12H cell. And then instead of coupling to the visual reporter, we couple it to a gas-producing enzyme. And then in this case, we had only the one microbe, so we were actually normalizing by cell number using respiration in this system. So what we found is that when you are comparing how much these microbes are sensing in terms of this, the bioavailability of this signaling molecule, it was very different when you were just using a liquid culture in the lab versus what you were when you were using a natural soil. However, we couldn't understand why. And here is when the artificial soil system came in handy, because you could design them to mimic different properties with a stacking complexity to mimic the natural soil, and we could get at these intermediate behaviors that could give us clues about what was happening and why the microbes were sensing more or less of this signaling molecule under these different environmental conditions. So what we are doing right now is that we're actually using these two technologies, the gas biosensors, the synthetic soils, to validate the traits that we are basically engineering using on automation systems, and as well as these different tools for QTL mapping and CRISPR-I. So here is a small example of how we are doing this. I mentioned velezensis before. We are using some strains, these FCB42 and GBO3, that you can find commercially available. We also have some natural isolates that we have isolated from the rhizosphere of poplar trees, which is what we use as our plant model system for bioenergy here at Oak Ridge National Laboratory. And this is an interesting system for microbiome engineering in general because you can use it for two different strategies. The first one is that these introduced bee biosensors can perform the beneficial functions themselves. So they can act as a soil probiotic and produce secondary metabolites that can help the plant. But the other strategy is that you can use these Bacillus velezensis strains to transfer beneficial functions into the rhizosphere, so acting as donors and then transferring this genetic cargo to the microbes that are already established in there. And why is it advantageous? A lot of the methods for horizontal gene transfer or genetic transfer within the rhizosphere, within the microbiomes in general, really, it's mediated by plasmids. However, biosensors uses another method that uses these ICES, or integrative or conjugated element mediated horizontal gene transfer, that basically integrates into the genome, and then it can be passively and stably propagated after several generations. So it's another mechanism by which you can engineer cargo into the rhizosphere directly. So everything coming together, basically we engineer QTL progeny of these velezensis, or we engineer these genome-wide CRISPR libraries into these microbial inoculant variants. And then we do large gene studies like that using next generation sequencing. And when we have traits that seem of interest to us, we engineer them with a constitutive gas biosensor or an inducible gas biosensor, depending on the trait. And then we can mix them with different soil types, soil amendments, synthetic soils to get at these mechanisms. And we can track over time how these different soil conditions impacts them, or if we add, for example, a nutrient pulse, whether if we can see some of these strains going back and continue performing. So this gives us a way of over time, several, I'm just showing here 7 days, but we have done some experiments that are longer, where we can track down again how these variants are behaving in the environment. And we have done this only with microbes and soil, and we are slowly moving towards also doing the exact same experiments with plant genotypes as well. So here I'm just showing a small picture of the plants in the different synthetic soils, and we are hoping that we can move toward larger scales in the future as well. So with that as a summary, I would like to just 3 key points to take away as home message. First of all, that genome-wide technologies are really useful for understanding microbial establishment and functional impact. Second, that gas biosensors are a new type of biosensors that allow for in situ real-time monitoring of microbial persistence in the environment. And third, that of course, the ultimate goal is to use natural soils and natural systems in order to study these microbes. However, synthetic soils are a good intermediate platform to get that mechanistic information and really understand how we can engineer these plants bioinoculants to act and be more productive and efficient. So thank you so much. Once again, if you guys are curious about this project, it has a lot more scientists that are working on it. We are also studying fungi. We are engineering plants. So you guys can check out the Secure Ecosystem Engineering and Design webpage. And I would like to thank all the team that works really hard on trying to understand these microbial inoculants. Thank you, and I'll be happy to answer any questions.
Thank you, Ilenne, for a very fascinating presentation. And you provided a lot of great information. And just for our participants, there were many links and resources that were discussed during the presentation, and all of those have been posted in chat. And unfortunately, our platform does not allow us to allow participants to copy the feature, but you are able to click on those links and then save them in that manner. So make sure you check out the chat panel before leaving today for those resources. So we do have a number of questions here that we're going to try to answer live. If for some reason that we do not get to all of the questions live, I think Ilenne has agreed that you may contact her directly if you have any questions that we're not able to get to live. So let's start, Ilenne, by talking about your different methods that you use to get this process started. You talked about QTLs, you talked about CRISPR. Can you talk a little bit more about kind of comparing and contrasting those different methods and kind of more about the results that you've seen? Has one method been better than the other, or does it depend? So can you talk more about that?
Yeah, absolutely. They are complementary. So again, one of the advantages of the CRISPR-I method is that you don't have you have controllable knockdown levels. So you can control the levels of how these genes are being expressed. So it's a little bit more of a subtle kind of like a change in terms of the regulation of that particular gene. So it really that one is exploring the regulation of the gene rather than the whole gene function, so to speak. However, with what we are seeing with the QTL is that one, if you have large fragments that get recombined, we can get at the genes itself that might be important. But also it allows you to study, for example, if different SNPs are important, which is not something that we can do with the CRISPR-I because we are just taking the whole gene and down-regulating it. So they are very complementary strategies. And we are seeing interesting effect with both. With the CRISPR, as I mentioned before, and there is a paper already published there that you guys can click on it. We have seen that there are some, when you upregulate a gene too much, it causes an effect that is deleterious. If you knock it down too much, it is also not good. So there is this regulation that you need to take into consideration. On the other hand, with the QTL, we have identified several candidate traits that are important. I showed germination there, but we have also are starting to understand, for example, biofilm formation and how complex it can be and how different ways that these microbes produce these exopolysaccharides affect how well they can behave in the environment. So they have done it for, once again, for germination, sporulation, for biofilm, for swarming, for swimming, which are two different ones. And we plan to explore, for example, secondary metabolite production as well. So a lot of different things to study with those two.
Okay, great, thank you. So our next question is about using GWAS and its approach with fungi. And would that be a promising approach for managing plant microbe interactions and desired outcomes? And what might this be more challenging than plant or bacterial GWAS shuffling approaches? So lots in that question.
Yeah, we are actually doing GWAS in fungi as well. We should be having a paper published soon. If you guys are interested, in the CDSFA webpage, there is some more information about those GWAS studies in fungi. So we are particularly interested in one pathogenic fungi. It's called Spirulina musiva and attacks poplar trees. And actually, the Bacillus velezensis strains that I work on are antagonists of these pathogenic fungi because they produce lipopeptides that can affect them. So yeah, we are also studying them from the fungal side as well. And we are also creating biosensors in the fungi to try to see how we can validate those traits as well.
So our next question is about the gas biosensors. Can you talk about or estimate what the cost might be to establish those?
You mean the cost in terms of what? Of like making them or
Yeah.
like having them
Yes.
as a technology for monitoring?
Or maybe having them as a technology for monitoring.
Yeah, so unfortunately, right now we just use these ones in constrained settings because these are engineered strains and we are not allowed them to just put them in the field right now with like the regulations that we have. So we just use them to again understand and try to discover new traits that are important for these plant bioinoculants. So I couldn't really tell how much it will be to deploy them in the field, like for farmers and things like that. It will be absolutely great. Even if we could have them like, I don't know, like tiny small vial where the farmer could like add some soil and then get a response really quickly, it will be really nice. But it's not something that we are thinking of right now. However, making the biosensors is extremely cheap. I don't know. I couldn't give an estimate, but maybe, I don't know, growing them, I don't know, really. But it's quite cheap technology.
Okay. Okay, thank you. Continuing on the biosensors, does the biosensor microbe interfere with the existing soil ecosystem?
Yeah, so two venues for that. So we have two different types of biosensors. I didn't go into details in this. I call them the spy and the blogger. So the spy is a microbe that is not present in that particular ecosystem, that we put them in there just to kind of like spy on, for example, the signaling that is happening between the native microbes. That microbe, if you are introducing it in there, of course, it can cause, for example, microbial abundance of shifts in the community, right? That's why we are trying to move towards trying to engineer these biosensors to be the blogger type kind of thing, where the microbes themselves that are in the environment are the ones that are reporting on what is going on in there. The production of the gas itself, yes, it can have some effects. It can have fitness on effects on the microbe that it's engineered, and it can have also maybe impacts in the overall community around it. And we are also trying to study how much that is, depending on case by case. So, for example, one of the biosensors that we are developing requires for us to add a little bit of salt in it. So we are trying to engineer them to be as sensitive as possible, trying to add as little sodium chloride as we can, because we know that it can cause stress on the plants and on the other microbes. But it's something that you have to take into consideration.
Great. Okay, so we have one participant who is working on a project with the objective to reestablish traditional rangeland grazing using the soil improvers that they are making. Looks like using biochar and other natural soil additives. And so, do you believe that the techniques that you discussed today could benefit such a project?
Oh yeah, I absolutely. So I actually did a lot of studies using these biosensors and biocharts as well. I don't know if that's the one that I'm particularly working on, but one of the things is that when you are using these soil amendments, there are a lot of different properties that can change. Let's take, for example, biochar. You know, pyrolysis temperature, the feedstock from which you are making the biochar, all those are going to affect the microbial community in different ways. And using these gas biosensors and the synthetic soils or whatever, we can start understanding the mechanisms by which is happening and maybe which variant of that biochart is better than others or what recipe it is. So we have used it in before actually for biochars, and we have also used it for woody chips to try to see which might be better one for some of the amendments that are required for different systems.
Hey, do you think that this research can be translated to a soil probe like tool for detection of specific microbes in a farm field?
Yes. Well, two things. We are so there are different technologies that you can apply it in the field. So for example, they have like these different like sortive cartridges that you can basically stack into the soil in the field, and then you can measure gases that way. So that's one way where the biosensor could be directly applied into the field and merged with that technology. The other one that we are trying, we are not trying it at field level, but we are trying it in the greenhouse. We have a really big plant phenotyping facility here at ORNL called Apple. And we use the online systems, right? So we just have, for example, the PTRMS connected to some of those plant pots, and then we can measure the gases that way. So we are hoping that that's something that we can do in the future, but might take a couple of extra years to develop.
Okay. And our last question that we have time for is one of our participants is curious about what your AHL detection results in liquid versus soil suggest about diffusion rates and spatial orientation of bacteria. Can you comment on this?
So one of the things that we discovered with that study, I didn't go into details about the mechanisms, but two things that were really, well, three things that were really important. First of all was the soil pH. So there was an effect in which the lactone ring that these molecules have pop open under alkaline conditions. So if you have alkaline soils, it will disrupt the communication between the microbes. So that was one of the things that we learned. The second thing that we learned is that depending on the signaling molecule that these microbes are using, for example, the one that I was using had a really long alkaline chain, it will get sorbed into some of the minerals. So then it would get stuck in there, and the microbes also got confused and couldn't communicate properly. So that was also something to take into consideration. And then the third thing that we learned was regarding drought and basically connectivity. And this is something that we want to study in the future a little bit more. So this is a diffusible molecule. So it requires, for water pockets to be interconnected so the different microbes can communicate with each other if they are, for example, in two different soil aggregates, right? Um, we hypothesized that when you are changing different hydration conditions, maybe they actually change the way that they are communicating too, and they use volatiles instead. So this is something that we want to explore a little bit further in the future too.
Well, wonderful. Thank you so much for your presentation today, your participation in this webinar, and we have a number of participants who joined me in their comments saying what a great presentation and an interesting topic this was. So thank you again for joining us and providing this great research, and thank you everyone for attending. We hope you have a wonderful day and evening.
Thank you. Thank you so much.